are gonna talk about the three organelles for today. Those are ribosome, endoplasmic reticulum, and Golgi apparatus. Okay? And for the assessment, guys, first 3 weeks topics you had to prepare. Okay? Well,
صار شنو؟ صار؟ اليوم عندك ورد؟
ايش قالت؟
ايه؟ هيك
ايش قالت؟
قالت جهزوا
okay.
آخر
Now
3
I
دروس
want
للأورال.
the complete quiet from you. Well, let's start from the ribosomes. Ribosomes are the smallest organelles actually, without membrane. Well, in ribosomes, what we have? 2 subunits we can see, large subunit and a small subunit. I want you to remember that the ribosomes in humans, in eukaryotes, are different than ribosomes in bacteria, in prokaryotes. Some of the antibiotics are targeting the ribosomes and preventing the protein synthesis, but it does not interrupt the protein synthesis in human cell because those ribosomal subunits are different, right? Mm-hmm. So the average size of the ribosomes are 20 power 30 nanometer. So we have a large subunit on the top and a small subunit on the basis, okay? So small subunit RNA is associated with 30 proteins to synthesize, and large subunit is associated with 15 proteins. So how the protein synthesis occurs between those subunits, right? Mm-hmm. From the 5' to the 3' prime. Okay. What are the function of ribosomes? The very important is that they are involved in the protein synthesis. Okay, protein synthesis without ribosomes is impossible, right? Ribosomes we can see as polyribosomes, as a chains of ribosomes, freely existed and present in the cytoplasm or associated with the endoplasmic reticulum. Sometimes those free ribosomes are going through the reticulum. After the protein synthesis, they leave the reticulum and go freely to the cytoplasm. Any other functions of the ribosomes are that they transcribe the messenger RNA. They simultaneously align the messenger RNA and the transport RNA, right? So ribosomes under the electron microscope appear as electrodense components, electrodense dots. We're gonna see them. But under the light microscope, the collection of the ribosome, we cannot see the individual ribosomes with a light microscope, right? It's impossible because of its size. But the collection of it creates what? Basophilic structure. Right? Because they have what? Ribosomic RNA. RNA has a phosphate group, negatively charged, so it will attach the basic state. Okay. Well, where are the ribosomes made from? From the nucleoli of the nucleus, because it contains the RNA. We are gonna learn the nucleus individually, but from the beginning, yes, remember that the compact portion, which we call the nucleoli, the center of the nucleus, contains RNA, where from the ribosomes are actually produced. And then, right, that's because the ribosomes are so small, they can easily pass through the pores of the nuclear membrane and then go through the endoplasmic reticulum or freely in the cytoplasm. And this is, guys, the electron micrograph, and you see, this is one ribosome, a few ribosomes, and these lineal original structure is endoplasmic reticulum with ribosomes on it. Okay, actually the function of free ribosomes and rebound polyribosomes are different. Let's see, here is a messenger RNA, it's 5' end and 3' end, and protein synthesis starts from the 5' and it goes through the small and large subunit, and amino acids are building up the protein chain, and free ribosomes are used to synthesize proteins of the cytosol and cytoskeleton for cell itself, right? And if some damage or something interrupts the protein synthesis, or protein is not synthesized properly, then it is metabolized and conjugated by protein which is called ubiquitin. Okay, and then proteosomes then degrade such damaged proteins. So the specific proteins for mitochondria, for peroxisomes, for nucleus, for the cellular organelles are building by the free ribosomes. What about the bound polyribosomes, which are present on the rough endoplasmic reticulum? They synthesize the ribosomes from exporting, right, to release them outside the cell. Then those proteins go through the Golgi apparatus, they are modified, they are packed into the vesicles, and then go outside the cell, or are used for the plasma membrane proteins. Okay. The same happens here if misfolding occurs or denaturation of protein. Proteosomes will degrade them. Just a second, I have to connect until it's. I hope the length will be enough. Okay. So now endoplasmic reticulum we can consider is one organelle, but there is a two type, rough endoplasmic reticulum and smooth endoplasmic reticulum. Actually the cell function is mostly determined which organelle is more leading organelle into the cell. For example, if the mitochondria is leading organelle, there's some electron transport is the main function of the cell. For example, the proximal convoluted tubule cells in kidney, they contain a lot of mitochondria. For example, the exocrine glands, like pancreatic acinar cells, they contain a lot of rough endoplasmic reticulum, so their major function is enzyme synthesis, right? For example, the liver cells, they contain a lot of smooth endoplasmic reticulum, so their function is the desintoxication, desintoxication and deamination also, there is a few important roles of the endoplasmic reticulum, including the detoxification or activation of foreign compounds, including the drugs that we talked about the liver, and also kidneys too. By endoplasmic reticulum, which is termed the cytochrome P450 proteins, and also storage of calcium. It's characteristic for the muscular fibers. Okay, especially in the skeletal muscle. In skeletal and cardiac muscle, smooth endoplasmic reticulum, lipids, synthesing of the lipids and protein is also the smooth endoplasmic function, but I'm trying to explain that smooth endoplasmic reticulum changes its shape and becomes a tubular. So in the and also the plasma membrane of the skeletal muscle fiber makes invagination, and it invaginates like a T-tubule, and either side of this T-tubule of the folding of plasma membrane there is a two cisterns of the smooth endoplasmic reticulum, and this structure we call triad. When we learn the skeletal muscle histology, we're gonna learn this again. In cardiac muscle, there is one T-tubule of plasma membrane and just one cistern of the smooth endoplasmic reticulum, and we call it dyad, right? Because it's two structures. Why it's important? Because from the plasma membrane, what goes through the cell? Impulse, right? And the smooth endoplasmic reticulum stores calcium ion. Calcium ion is very important for the muscle contraction, because once the calcium ion binds to the troponin, it will tropomyosin. It will free the troponin. It will free the actin to bind to the myosin head, and that's initiation of the muscular contraction. Without calcium ions, there won't be muscular contraction, right? Okay. Let's see again the functions of endoplasmic reticulum. Like you said, synthesis of the proteins and lipids also. It's a place of the chemical reaction. Smooth endoplasmic reticulum, especially detoxification of drugs and some other chemicals like alcohol, and this is a system of proteins which we call P450. Okay, so storage of the calcium and also newly synthesized molecules, and transport. Some of the molecules move through the cisternal space from one part of the cell to another. So the cisterns of the smooth endoplasmic reticulum can be used as a transport of the molecules inside the cell. Okay, this is a rough endoplasmic reticulum. Again, remember guys that rough endoplasmic reticulum has a linear structure, and the smooth endoplasmic reticulum mostly have tubular structure. And also rough endoplasmic reticulum appears dense, electrodense under the electron microscope. You see the polyribosomes on the membrane. Its membrane, the similar as plasma membrane, right? This is what type of image? Fluorescence. Immunofluorescence method is used for this image, and target is the membrane proteins of the endoplasmic reticulum, and it gets green color. So in this immunofluorescence staining method, green is revealing the endoplasmic reticulum, and this yellow reveals mitochondria. We will learn mitochondria later. This is the relationship between rough and smooth endoplasmic reticulum. You see, smooth endoplasmic reticulum transport vesicles to the Golgi apparatus, and then here we have tubular cisterns, and their surface is smooth without ribosomes. That's it. Okay, how the protein synthesis occur on the rough endoplasmic reticulum. But I want you to pay attention that on the surface of rough endoplasmic reticulum there is a receptor protein and there is core protein. Okay, what is receptor protein necessary for? Because it binds to the ribosome and it binds the messenger RNA with ribosome. Okay, and then when the protein amino acid chain is building, look, this peptide is binded through the membrane pore. Okay? Once the peptide is synthesized, then it is removed, and ribosomal subunits are also removed from the surface of the rough endoplasmic reticulum. Also remember those receptor proteins and membrane pore. So in this text I have the same, free cytosolic ribosomes attach to the messenger RNA and begin to produce peptide. That's the A. This is a red one is messenger RNA. And here the ribosomes attach to the receptor of endoplasmic reticulum membrane, and the peptide is threaded into the endoplasmic reticulum lumen by protein pore. That's channel. Like a transmembrane proteins we have in the plasma membrane. The C1 is when the peptide is synthesized, when the original signal sequence that threads the peptide into the endoplasmic reticulum lumen is cleaved, the peptide is made, its forms in lumen. See? Then synthesized peptide is go through the lumen of the cisternal endoplasmic reticulum. And then after completion of peptide synthesis, the ribosome detaches from the receptor protein, right? And then returns to the cytosolic free pool. That's the same actually. That's transport RNA and messenger RNA. They assemble in the ribosomes simultaneously. Ribosomes have a signal recognition particle which binds to this receptor. Okay? And there is a signal recognition receptor, particle receptor. And that's how ribosomes attach to the rough endoplasmic reticulum. And that's a pore protein translocator complex. When the growing chain of the polypeptide is done, completed polypeptide, it goes through the lumen of the rough endoplasmic reticulum, and the ribosome is detached from the endoplasmic reticulum. Well, what about the smooth endoplasmic reticulum? Now separately, let's talk about it. The smooth endoplasmic reticulum is a site of membrane lipid synthesis and protein processing. Okay? It has three main functions. First of all, enzymes in the smooth endoplasmic reticulum perform the synthesis of the phospholipids and steroids, and those are the major components of the cell membrane, right? So they are produced from the smooth endoplasmic reticulum. Other enzymes, like we mentioned, cytochrome P450 family, they allow detoxification of harmful exogenous molecules such as alcohol, such as some chemicals, such as some drugs, barbiturates, and mostly they are present in liver cells. Those enzymes also produce some component of bile. Okay? The hemoglobin degradation product, bilirubin, is also undergoes degradation by the smooth endoplasmic reticulum. When the liver cells are damaged and the smooth endoplasmic reticulum is damaged in liver cell, the bilirubin is not degraded, and it causes what? Jaundice. Yellowish of the skin and sclera, right? And itching sensation. Sometimes it's caused by some viral or autoimmune process in the liver that damages the liver cells itself, or it's genetically determined for newborns. Newborn jaundice can be caused by the defect of the smooth endoplasmic reticulum inside the liver cells. Okay, smooth endoplasmic reticulum vesicles are also responsible of sequestration and controlled release of the calcium. We have already mentioned this in muscular cells. What does sarcoplasmic reticulum mean? When we will learn the muscular tissue, we are gonna meet this term again. Sarcoplasmic reticulum is the same as smooth endoplasmic reticulum, but in the muscular fiber. Okay, now before we talk about the Golgi apparatus, I want to see that cells are very different. Cells might have a different appearance according, as we have already mentioned from the beginning, according which organelle is leading and which organelle is more prominent and more developed. For example, erythroblast. Erythroblast is a primordial stem cell of the erythrocyte. Erythrocytes we also call as red blood cells. But look at it. Is it red? No. Why? What happens here? Nucleus is purple color, you see, and cytoplasm is blue. And why? Because erythroblast starts synthesizing the hemoglobin, right? Before the hemoglobin is made, we have a lot of free ribosomes here and endoplasmic reticulum, which makes the blue color of the cytoplasm, right? That differentiates, this is a different staining method, that's ordinary erythrocytes. They are not red because it's a different staining method. But we can clearly see the basophilia of the cytoplasm of this cell. Well, eosinophilic leukocyte, that's a major blood cell. See what we have? Red dots inside it, eosinophilic granules. Why they are eosinophilic? Because they contain the proteins, chemotoxic factors, and other membrane. Sorry. Excuse me. So this characteristic appearance helps us to differentiate and identify those cells. And by the way, eosinophils have bilobed nucleus. See? This is not many nucleus. It's lobed. And small bridges, thin bridge, connect those lobes with each other. Plasma cell is modified B lymphocyte, and leading organelle here is two organelles, rough endoplasmic reticulum and Golgi apparatus. Here you see nucleus, right? And you see the darker area?
Yes.
That's rough endoplasmic reticulum, but between the nucleus and the rough endoplasmic reticulum, there is pale area. That this is a Golgi apparatus plate there. And it gives a specific appearance. We're gonna meet those cells again and again. It's just for illustration. Okay? Now let's see pancreatic acinar cells, the secretory cells which have a lot of secretory granules. Look at it. The basal part of the cell contains nucleus, a little bit darker cytoplasm because of rough endoplasmic reticulum, and lot of granules. Secretory granules. Secretory granules is the next week's topic. I have not included it to my presentation, but just to show you. And this is jaundice, how the scleral jaundice appears. So, yellowish discoloration of the skin is caused by accumulation of bilirubin and other pigmented components. If the bilirubin is not degraded and metabolized by smooth endoplasmic reticulum, it might be caused by damage of the liver cells by hepatitis, right? You might have A hepatitis, B, C, right? Major symptom is a yellowish of skin, right? Which we can observe on the skin. And this is because the smooth liver cells are damaged, smooth endoplasmic reticulum does not work properly.